For 50 years, it was one of the biggest puzzles in all of biology, a secret code that life itself uses to build its most essential microscopic machines. And then, almost out of nowhere, an AI solved it. Today, we are going deep into that mystery, the problem of protein folding. Okay, just for a second, think about this. Right now, as you're listening to my voice, trillions, and I mean trillions, of tiny, intricate machines are buzzing away inside you. They are the microscopic architects and engineers that are literally running the show. So, what are these miniature machines, these incredible little workhorses of life? Well, you definitely know them by their more common name, proteins. I mean, they do everything. One type of protein is grabbing oxygen and carrying it through your blood. Another one is acting like a tiny pair of scissors chopping up the food you eat. And a different kind is your personal bodyguard hunting down and fighting off viruses. Pretty much nothing happens in your body without them. But here's the crazy part. Every single one of these complex, powerful machines starts its life in a surprisingly simple and, frankly, totally useless form. And that right there is what leads us to the great folding mystery, a problem that stumped scientists for half a century. You see, every protein begins as just a long, floppy string of molecules called amino acids. The best way to picture it is like a necklace, made from maybe 20 different kinds of beads. In this state, it's just a string. It can't do a thing. It can't fight a virus, it can't carry oxygen. It's like a robot that's still in the box in a million pieces. So the billion-dollar question is this. How does this long, floppy, useless string manage to transform itself into a perfectly structured, three-dimensional working machine? And the answer is a process that honestly sounds like something out of science fiction. It's called protein folding. In a fraction of a second, that string just spontaneously collapses and twists into a perfect, intricate 3D shape. It's like a piece of high-tech origami that folds itself. Now, why are we so completely obsessed with this final shape? Well, because if that folding process goes wrong, even just a little bit, the consequences for our health can be absolutely devastating. There's this golden rule in biology, and it's super important. Shape determines function. The final 3D structure of a protein is everything. It's what dictates exactly what job that protein can do. Think of it like this. A correctly folded protein is like a perfect key. It's been precision engineered to fit one specific molecular lock in your body. But if it misfolds, well, now the key is bent. It's useless. And it gets worse. These bent keys can get all sticky and start clumping together. And these toxic clumps of misfolded proteins, we now believe they're a key factor in what builds up in the brain and contributes to terrible diseases like Alzheimer's and Parkinson's. So yeah, figuring out folding isn't just a fun scientific puzzle, it's absolutely critical for finding cures. For 50 years, predicting a protein's final shape just from its amino acid string was seen as one of the grandest challenges in all of biology. And that's because the sheer scale of the problem, it was just mind-boggling. Okay, so this gets to the heart of what's known as Levinthal's paradox. For just a single, average-sized protein, the number of possible ways it could theoretically fold is, well, it's astronomical. We're talking about a number with more zeros than all the atoms in the entire known universe. No joke. So get this. If a protein had to find its correct shape by just randomly trying out every single one of those possibilities, it would take longer than the current age of the universe. And yet, inside your body, it happens in microseconds, a blink of an eye. How is that even possible? This is the question that baffled an entire generation of scientists. Scientists chipped away at this for decades with really slow, painstaking lab work. But the massive breakthrough, the thing that cracked the whole code, it didn't come from biology at all. It came from artificial intelligence. Researchers at a company called DeepMind created an AI named AlphaFold, and what they did was brilliant. First, they sent it to school. They fed it over 170,000 protein structures that we already knew. You can think of it like a detective studying thousands and thousands of old case files. Then, using a technique called deep learning, AlphaFold started to see the hidden patterns, the secret rules of how proteins fold. It learned which little beads on the necklace like to be near each other. And finally, it was ready for the test. You could give it a brand new, unknown sequence, and AlphaFold could predict its final 3D structure with unbelievable accuracy. This wasn't just an improvement, it completely changed the game. I mean, a task that used to take a PhD student years of their life and tons of expensive lab work could now be done in minutes on a computer. Just making AlphaFold was incredible, but what the team behind it did next, you could argue that was even more revolutionary. See, they didn't just keep this amazing new tool for themselves. They put it to work. They used AlphaFold to predict the structures of over 200 million proteins. That's nearly every protein known to science, from basically every organism on Earth. And then, they just gave it all away. They put every single one of those structures into a public database, online, completely free, for any researcher anywhere in the world to use. They essentially published a library of life. Now, any scientist, whether they're in a tiny lab or a huge research institute, has the map to life's machinery right at their fingertips. And the world absolutely took notice. In 2024, the leaders of the DeepMind team were awarded the Nobel Prize in chemistry. And the reason? The official citation said it all. They were honored for solving one of the grandest challenges in biology. So the great folding mystery is, for the most part, solved. We now have the blueprints, the actual instruction manuals for the tiny machines that run the entire living world. And this opens up incredible new doors for designing drugs, for understanding diseases, even for creating brand new proteins that have never existed before. The only question left now is, what are we going to build with them?